Welcome to Spotlight on Antibodies, a podcast from BioIntron where we bring you the latest antibody industry trends. I'm your host, Jovina Celestio, and this month in September, we'll highlight recent approvals in biosimilar drugs, conjugate linker designs, and discovery platform advances. As always, links to sources are in the episode description. Monoclonal antibodies for clinical use typically have high efficacy and favorable tolerability due to selective antigen targeting. However, their complex structure, especially as glycoproteins produced in diverse cellular systems, makes them expensive to manufacture, limiting patient access. These cost and accessibility challenges have fueled the development of biosimilars, biologic products designed to be copies of the reference mAb, but are often slightly different in pharmacodynamic and pharmacokinetic properties because of changes in glycosylation. In response to escalating healthcare costs, various global markets have implemented policies to promote biosimilar adoption. For example, Japan mandates an initial biosimilar pricing at 70% of the reference product. Despite this, uptake remains variable, as biosimilar replacement rates range widely, from 90% for filgrastim to just 20% for infliximab. Contributing factors include limited clinical data for minor indications, inconsistent quality information compared to small molecule generics, and inadequate understanding among healthcare providers and patients. In contrast, regions such as the EU have achieved higher biosimilar penetration through regulatory support and prescriber autonomy. France, for instance, allows automatic substitution of TNF-alpha inhibitors unless explicitly restricted, resulting in replacement rates nearing 80%. These past few weeks, multiple biosimilars have been approved or advanced by regulatory agencies across the US, Canada, and Europe, with denosumab emerging as a dominant target molecule. The US FDA recently approved multiple denosumab biosimilars, Bildios and Bilprevda from Henlius and Organon, and Bosaya and Alkelso from Biocon Biologics. All four products are approved for the full range of indications of the reference products, Prolia and Xgeva. Notably, the Biocon products received provisional interchangeability status, a regulatory designation that may encourage greater formulary uptake. In Canada, Celltrion's Stobaclo and Ozempic also secured approval for all Prolia and Xgeva indications, supported by robust Phase 3 data. In Europe, the EMA has issued positive CHMP opinions for both Alvotech's AVT03, a denosumab biosimilar, and Gobivaz, a golimumab biosimilar. Meanwhile, earlier this month, a groundbreaking milestone for pharmaceutical innovation was achieved with the announcement of the first ever FDA acceptance to waive clinical efficacy studies for monoclonal antibody biosimilars. It was argued that clinical efficacy studies add no scientific value to biosimilar evaluation, as analytical similarity and immunogenicity studies are sufficient to confirm biosimilarity. Thus, the biosimilar application for Stelara will be the first filed with the FDA without requiring any clinical testing, marking a precedent-setting regulatory breakthrough. Next up, we'll talk about considerations in ADC linker design through stability, efficacy, and precision. The current generation of approved antibody-drug conjugates reflect a broad spectrum of linker chemistries, with cleavable linkers, particularly peptide-based systems, predominating. These linkers are typically cleaved by tumor-associated enzymes such as cathepsin B, plasmin, or legumain, enabling selective drug release within the tumor microenvironment. The design of such peptide linkers has centered on optimizing their protease sensitivity while maintaining adequate stability in systemic circulation. Despite the clinical success of this strategy, limitations remain, including premature release, low drug-to-antibody ratios, DARs, and hydrophobicity-induced aggregation. Advances in peptide linker designs through chemical modifications and structural adaptations include para-aminobenzyl modifications, often used as self-immolative spacers, which can be re-engineered into exocleavable linkers, or glycopeptidic linkers, for hydrophilicity and enzymatic sensitivity. Additionally, the incorporation of hydrophilic units has proven effective in mitigating aggregation and improving circulatory stability. Another advancement involves the introduction of an exolinker configuration, wherein the cleavable peptide sequence is repositioned to the exo site of the p-aminobenzyl carbamate moiety. This incorporates hydrophilic amino acids such as glutamic acid to improve aqueous solubility and reduce aggregation, even in the presence of hydrophobic payloads. Preclinical evaluations demonstrated that this design significantly improves the in vivo stability of the conjugate and has high DARs. Linker design has influenced ADC pharmacodynamics, particularly through the bystander effect, which enables the diffusion of released cytotoxic agents into neighboring cells, including those with low or absent antigen expression, thus expanding the therapeutic footprint of the ADC. However, this mechanism is heavily modulated by the physicochemical properties of both the payload and the linker. Computational studies have shown that the linker contributes substantially to the overall molecular size and hydrophobicity of the drug-linker complex, impacting its ability to permeate cellular membranes. Notably, payloads with high ionization states face additional membrane diffusion barriers, limiting their capacity for bystander activity. These findings suggest that linker design must consider not only cleavage and stability, but also the biophysical properties that determine tissue penetration and intracellular trafficking. Now, computational platforms play a pivotal role in streamlining ADC development. Molecular modeling and AI-assisted structure prediction are being used to assess antibody linker payload compatibility, predict degradation pathways, and simulate drug release kinetics. These tools enhance early-stage candidate selection, reduce the need for iterative in vitro testing, and facilitate the rational design of linker architectures with finely tuned stability and release profiles. Overall, the ideal linker system must be stable in plasma, selectively cleaved at the tumor site, compatible with diverse payload chemistries, and inert to biological interferences. The final topic of this discussion are novel platforms for antibody discovery. High-throughput technologies are reshaping the antibody discovery landscape, with platforms now capable of rapidly producing highly specific, naturally paired antibodies. Antibody library display systems, such as phage, yeast, bacterial, and ribosomal display, have matured through the integration of automation, next-generation sequencing, and microfluidics, enabling faster enrichment and broader repertoire recovery. In parallel, single-cell isolation methods, such as droplet and microwell-based microfluidics, now enable the functional screening of millions of antibody-secreting cells with unprecedented precision. Emerging innovations suggest that the integration of high-throughput biophysical, cellular, and computational technologies are soon to come. Here are some examples published over the past month. First, the OPAL Plus system combines large-scale antibody production with parallelized binding assays, reducing the time for evaluating antibody-antigen interactions. In its first application, researchers profiled 300 influenza hemagglutinin antibodies, uncovering shared binding features across diverse immune repertoires. Importantly, OPAL Plus reduces material costs by up to 90% and shortens experimental timelines from months to days. Beyond influenza, the platform is poised to rapidly identify therapeutic antibodies against emerging pathogens and to serve as a validation engine for AI-based antibody design models. A second example is HT-NABs, a silicon nanophotonic and acoustic bioprinting-enabled platform capable of simultaneously screening antibody-antigen interactions with picomolar sensitivity. With independently functionalized nanosensors, the system quantitatively measures binding kinetics, affinity constants, and specificity. demonstrated on antibodies against SARS-CoV-2, influenza, and therapeutic EGFR targets, HT-NAbs enables not only rapid affinity screening but also epitope binning, ensuring broad repertoire diversity. Their design marks a major step towards bridging computational protein design with empirical validation. A third workflow integrates pan B-cell enrichment with droplet microfluidics to overcome the long-standing challenge of limited markers for rabbit B cells. By using magnetic negative selection and tailored antibody cocktails, researchers achieved a 7-fold enrichment in IgG secretion, enabling high-throughput screening of rabbit antibody repertoires. Droplet-based functional assays for both soluble and membrane antigens preserved natural VH and VL pairing, and yielded antibodies with binding rates compared to previous reports. The approach accelerates rabbit mAb discovery, expanding their potential in diagnostics and precision therapeutics. And that's all for this month's Antibody Industry Trends. To learn more insights into antibody drug news, make sure to follow BioIntron on LinkedIn, on our website at www.biointron.com. Keep up to date with further episodes from Spotlight on Antibodies by following this podcast.